Good morning. I'm Christine Chalk. I'm the program manager for the CSGF at the U.S. Department of Energy's Office of Science, and I'm happy to moderate the first panel this morning. Our first speaker is Alnur Ali from Carnegie Mellon University. His work is in the field of machine learning. His advisor is Zico Coulter, and his practicum is at the Lawrence Berkeley National Laboratory.
Great, thanks. Okay, good. Yeah, thanks for coming. So the title of my talk today is, actually, can people hear me okay? Okay. Is Distributed Sparse Inverse Covariance Estimation from fMRI data for segmenting the human brain. So I'll unpack a lot of that later in the talk, but maybe just to get things started, a quick footnote. So the content I'll cover is actually based on our recent paper. I'll put the title down here, which actually grew out of work I did in my practicum a few summers ago. So if people are interested in learning more about that or want to point to the paper, feel free to find me later. I'm happy to point you in the right direction. OK, so just by way of an outline, so I work in statistics and machine learning. So I figured, I'm not sure how many people here do the same, but I figured it might be a good idea to at least spend kind of a decent amount of time going over what exactly sparse inverse covariance estimation is. Then I'll talk about our computational approach, so specifically the algorithm we use for doing sparse inverse covariance estimation. Then I'm going to go through an illustration of how we took our method and applied it to the fairly open-ended problem of how to understand how different parts of the human brain work together. Then I'll wrap up just by kind of looking back over everything that I discussed. OK, so sparse inverse covariance estimation. So this is actually a topic that's kind of long-standing interest to statisticians. And the way I kind of find it helpful to think about it is just to kind of break it down piece by piece. OK, so covariance estimation, maybe that's a good starting point. And maybe that's a little bit more natural to people here. So covariance estimation, so that basically just means there's a covariance matrix associated with a collection of random variables that we want to estimate. So maybe a little bit more mathematically. So suppose we observe n samples of a p-dimensional Gaussian random vector that has some unknown but underlying covariance matrix here denoted as sigma naught. So the task at hand is we want to estimate. We want to go ahead and estimate this guy, sigma naught. OK, so what do we do? So the kind of common answer to that is try maximum likelihood estimation. So maybe to just kind of remind people how that works, so maximum likelihood estimation just boils down to 3 steps usually. It's step number 1, you write down, in this case, the Gaussian probability density of a sample. And that's going to depend in some unknown way on this unknown parameter of interest. That's sigma naught. We take logarithms. That just sort of makes the math work out easier. And then we maximize. Or conversely, we minimize kind of minus the expression we're working with. OK, so maybe you do all that, and actually it just boils down to this expression I've written here. This is an optimization problem. Maybe it looks a little bit cryptic, but I'll work through the symbols. So what we want to do, if we're given a bunch of n samples of a p-dimensional Gaussian random vector, and we want to estimate the underlying but unknown covariance matrix, what we do is we minimize. Here the variable is denoted sigma. We minimize over all sigma constrained to lie in. This is the space of p by p positive definite matrices. The log determinant of sigma plus the trace of the sample covariance matrix times sigma inverse. I don't know, maybe that sounds kind of crazy. I don't know, but that is what it is. You just kind of work out the math. That's what boils out. And if you stare at that thing long enough, you sort of realize that there's a problem, which is that this optimization problem is actually not convex. It's not convex in the variable of interest, which again is sigma. So why is that a problem? Well, typically in a lot of fields, statistics and signal processing and a lot of other fields, convexity is sort of a good thing. It means our algorithms will sort of converge fast. It means that they'll converge furthermore to a solution that's globally optimal. So that's kind of a thing we look for. The point is, if we want to do covariance estimation, this is the problem we solve. And there's immediately kind of a hurdle. That's at least the message, in that this problem's not convex. OK, so you might just kind of stand up and say, well, I don't know, who cares about that? Like, we're doing covariance estimation. Why don't, like, how about the sample covariance matrix? We know what that is. We can kind of write it down. I've written it down here. And so just sort of in terms of the notation, because I'm going to revisit some of these symbols later, X here is what's called in statistics the design matrix or the data matrix. It's an n by p matrix. N rows, one for each sample. P columns, one for each dimension or variable or feature or whatever terminology you like. And you might say, well, we can just form this guy here, and that should be maybe a good estimate of the underlying covariance matrix. I guess the other problem is, if you stare at this guy long enough, if you have more features, so here I've denoted that as P, than samples, this guy turns out to be singular. Okay, so like that's a problem if you want to, of course, invert it. Or like, for example, if you want to stick, we talked about Gaussian densities before. If you want to maybe estimate this guy, stick it back into Gaussian probability density and use that to maybe make predictions, like that's not going to work. Okay, there's a third problem, and this one's maybe a little bit kind of more intricate, so I'm going to have to be necessarily vague, which is that the sample covariance matrix, so this guy, if you say you picked some entry of it, and you looked at the corresponding entry at the population level, so that was sigma naught, that was denoted like a couple slides ago. Look at the corresponding entry. Those two entries are going to be like very far away. So I guess maybe put a little bit more crudely, the sample covariance matrix actually turns out to be like a poor or like a bad estimator of the underlying covariance matrix. And that, again, if p is greater than n, so if you have more features than samples. So I guess the upshot of all this is you might think like, okay, if we're interested in covariance estimation, why don't we just use the sample covariance matrix, like dead end kind of thing. That's basically the message. Okay, so solutions. So what do we do? There's a simple fix for problem number 1, which is basically about the non-convex, sort of the optimization problems not convex. The fix is you just simply change variables. So instead of before we were minimizing over sort of the space of covariance matrices, now we're going to actually work with inverse covariance matrices. So I'm denoting that as omega here. So that's just that before the covariance matrix is sigma, now just sigma inverse, and we're just going to call that omega. So the optimization problem kind of boils down to now minimize over omega, constrained again to line the space of positive definite matrices, minus log det plus trace, again, the big S here is the sample covariance matrix, times omega. So sample covariance matrix is here, but it's not the variable anymore. It's just sort of like problem data that figures into the problem. The variable is the inverse covariance matrix. It's omega. So this guy turns out to be convex. That's the long story short, and so we can kind of check that box. So problem 1 in the clear. Problem 2, problems 2 and 3, which are basically about this sample covariance matrix being like a poor estimator. So the fix there is a little bit more subtle, and the solution is we're going to add regularization. So what I mean by that is specifically we're going to solve, so far this part of the problem, minimize minus log det trace. That's the same as the thing I just walked us through. But now we're going to tack on this term in purple here, which is a regularization term. And specifically, it's the element-wise L1 norm of the variable omega, which just means you sum up the absolute values of the entries of omega. So like I said, this thing's a little bit more subtle. Maybe it's clear to you why this is a reasonable thing to do, maybe not. But the long and the short of it is that actually fixes those second and third problems. And we're still convex. So I guess the message on this slide is, so hopefully it's clear actually by now. So the inverse covariance estimation part of the overall kind of title, like we're basically doing inverse covariance estimation, right? So hopefully that's clear. The sparse part, maybe not, maybe that's a little bit fuzzy. So I'm going to try to get to that on the next slide. Okay, so there's another kind of benefit of that L1 penalty that I was just talking about, and I think it's not totally obvious. So the benefit is this. So if you solve this optimization problem, right, this is our like inverse covariance estimation problem. So you get out omega, right? So that's a matrix. It turns out you can take that matrix and turn it into a graph, okay? So the way that works is for each variable, you draw a node. and then you don't put an edge between nodes if their corresponding entry in the inverse covariance matrix is 0. So I don't know, maybe like a picture might help. So I kind of put down here like a toy inverse covariance matrix. MG, I don't know, these are like synthetic labels I made up for the different variables. But you can see I have vertices for MG and A, but I didn't put an edge between M and A because their corresponding entry had a 0. So inverse covariance matrix goes to graph. That, I don't know, maybe that seems silly, but actually this kind of thing is pretty useful for exploratory data analysis. Like people do this all the time, like they actually use this thing in biology and finance. So this is actually kind of useful just to get a sense of like what is happening in the data, or the interpretations loosely that maybe these M and G variables and the G and A variables, they interact somehow. There's some sort of relationship between those, but not between M and A. So this is actually kind of a useful thing. I guess furthermore, the L1 regularization penalty, man, I got to pick up the pace here, is actually gives us a sparse graph. So what that turns out to mean is we actually get a graph with not a lot of edges. Okay, so hopefully that clarifies sparse inverse covariance estimation. All right, in my quest to pick up the pace, so the computational, so enough background. So what do we actually do? So our kind of like interest is to solve the sparse inverse covariance estimation problem. So that's minimize over omega, constrained to line the set of positive definite matrices minus log det plus trace, and then plus that regularization term. So what do people do to solve that thing? A common approach is to use what's called a proximal gradient method. You see that all over the place in the machine learning literature. It's related to, but definitely different from an implicit Euler discretization of the gradient flow. I don't know if that makes sense to people here. So that's what you do. And if you kind of work it out, there's two computational bottlenecks, and they both involve multiplying dense matrices together and dense and sparse matrices together. And so those bottlenecks are actually really bad if you have a lot of features. And so to the extent that you can't actually solve this problem on a single machine. So we actually use a distributed approach. In particular, we use this class of distributed algorithms called communication avoiding algorithms, which people at Berkeley Lab know a lot about, which is where I did my practicum, to actually kind of multiply these matrices, these dense and dense sparse matrices together in a distributed way. So that gave us like a huge computational boost, and that made our method kind of scale to multiple machines and big data sets and work really fast. Definitely don't have time to go through an example, but feel free to like grab me later and I can walk you through it. I'm going to try to skate through the empirical evaluation, which is maybe looking back. So we've got this method we developed. The claim is you give it a data set that's like n rows, one for each sample again, p columns, one for each variable or each feature, and it returns to you a sparse estimate of the inverse covariance matrix. And from that, you can generate graphs and all sorts of things. So we said, okay, what's a big data set out there? Because our method can scale to big data. And so we looked at neuroscience. And so we took our method and tried to make progress on a challenging problem in neuroscience, which is understanding which parts of the brain work together. Okay, so our starting point for that investigation is an fMRI data set from the Human Connectome project. So that's like a recent NIH research initiative. And so the data takes the form of, again, n rows, where you can roughly think of each row as corresponding to a different patient that was put inside an fMRI machine. And p columns, where each one of the p columns corresponds to like a spot on the patient's cerebral cortex. So that actually turned out to be a pretty massive data set. The number of columns, p here, that's the number of features essentially. It was about 91,000. And it turns out like a lot of methods choke on that. So our method did pretty okay. And so by our method, I mean specifically, we take that data set I just talked about, run our method on it for sparse inverse covariance estimation, get a graph out, and then we actually cluster the graph. So we're going to actually basically segment the graph into connected components, and the interpretation is each of those connected components is sort of like a functionally coherent region of the brain. By the way, we just to maybe shout out NERSC and Edison, we like ran all this on NERSC and Edison, and that was like a huge lifesaver. It was, it was easy to get running, and it worked great. Okay, so just to go through the results, so we compared. So actually, our method is on the bottom row here. The top row is a baseline. It's a pretty recently published paper in Nature. And so I guess the distinction between the baseline method and ours is our method's basically totally data-driven, right? All I said was you give it a bunch of data, it returns to you like a segmentation of the brain. The top method actually was like, it's quite labor-intensive to generate. There's some algorithmics, but it's kind of very much done by hand. And so the different colors here, I've split the clusterings into the left and right hemisphere. And actually, the colorings don't really like even have to match up between the different plots. They just sort of like section off clusters. And so I guess, maybe at a 20,000 foot level, you might say that like, I mean, this isn't the worst thing I've ever seen, right? Like, you sort of look at this plot here. So from a 20,000 foot level, like, there's a red patch here, which our method deems to be a cluster. There's kind of like a strip up here, which the baseline says is a cluster. And you can sort of play the same game on the right-hand side. But in a little bit more detail, so if you actually go through and look at some areas that are of interest to neuroscientists, so for example, areas that are thought to be useful in terms of hearing or sort of eye movement or sensory processing, if you actually kind of look closely, so for example, area 55B thought to be involved in hearing. So that's right here. So the baseline gets it. And actually, I mean, it might be hard to kind of see from the back, but like it does look like our method is able to pick that up as well. There are certainly things that our method does not do well at, but I would say as a first pass, and that's kind of how I'm treating these results, our method seems to do a reasonable job, especially given like no hand tuning at all. And it certainly did better than some baselines. Okay, and just to wrap up. So talked about a method for sparse inverse covariance estimation. Hopefully that problem setup made a little bit more sense than the beginning of the talk. If not, I'm happy to clarify later. Our method, and I didn't have time to get into this, is a lot more scalable than other methods in the literature, so it's able to scale to big data. I did illustrate an application of our method to trying to uncover structure in the brain. And I guess the neat thing is our method was able to do that without kind of being told how. So it's just entirely data-driven thing. All right, that's it. Thanks.